So, hello everyone again. Welcome to another Saturday microscopy live stream. Microbehunter here. Hope that you're doing fine and I hope that everyone's able to hear me properly. And yes, you can always, of course, post a question if or a comment if there are some audio problems because I used to have some audio problems some time ago. Well, what am I going to do today? Well, today I would like to look at a few microscope slides that I've got here, permanent slides. These are commercial permanent slides. Hi, yes, hello, everyone's commenting right now. Okay, great, seems to work. Yes, I'd like to look at a few commercial permanent slides today. I've got a few here, I've chose a few of them. And primarily I would like to look at mammalian tissue, so tissue from mammals. I used to call, up to about 1 hour ago, I used to call the episode, the title, Body Parts Under the Microscope, and then I realized this kind of sounds a little bit strange, so I changed it around to tissues under the microscope. Some of the tissues here are from humans and others are from other animals, but it doesn't matter because it looks exactly the same from also among all mammals. Okay, so what I would like to do is I would like to simply show them to you and comment a little bit and hope that you consider them interesting. I need to give you a short background before I start. A short background information here. I just recently received a comment beneath one of my YouTube videos where somebody said that that they like the channel very much and it's all very interesting, but it's a little bit repetitive because I'm always putting the same specimens under the microscope, like of course plants and water samples, and I agree. It is becoming a little bit repetitive also for myself, and there has been the request therefore maybe to also put some animal tissues, also human tissues under the microscope. And because I do not make human tissues myself with permanent slides, I just want to show you some commercial slides that I've got here. A little bit of background here because I know that I receive occasionally also questions on where can you buy those slides. With the simplest answer would be you can buy slide boxes like this even from Amazon. Even they have it. The quality varies, I have to say. The other slides that I have over here is I've got from a German company who are basically making them for educational, for education, but they're selling things, slides like this, all over the world as well. But I say German because in case you see the labels here, they're all in German. Because I'm a teacher in a school and therefore I was able to obtain them as well. So what I'm going to do is I'm simply going to show you a few things here I'm going to switch over to the microscope view, and if you have any questions, of course you're welcome to ask them. No guarantee that I'm able to respond to all of those questions but I'll try my best. And there's one thing that's a little bit new this time and look, what do I have over here? There's an arrow. And I'm able to now move an arrow around to point towards the different parts. And I think this makes life a lot easier here. And I already have the first specimen here under the microscope using the objective over here. That's the 60 times objective. And this is so-called compact bone. That's bone. What we have over here is we have over here bone under the microscope, and one can already see that the bone is not homogeneous, but is somehow organized and structured. And there are those circles over here, and there are those black dots over here. And those black dots here, these are bone cells. Okay, so there are cells in bone, and this is the so-called diversion canal, and this is where also blood is transported. So there are blood vessels inside the bone, and there are also bone cells, and you can also see the rings that you see here, these are called lamellae, what you cannot see so well here, you have, it's very fine, a little bit better over here. There are also certain canals, which transport the blood, like this outwards. Do you know staining techniques used? Okay, that is a problem. It's a very good question, and something that I have to tell you annoys me a little bit is because the slide does not actually mention this. Okay, so I'm going to show you here. Okay, it says here bone from a human bone. It's a compact bone cross section. and that's it. Yeah, so it does not tell me anything about how it was processed, which I personally consider a little bit of a pity, because there's so much space here on the right side where you put the number. You could actually make this provide some valuable information here as well, but it doesn't say that. It says only bone from the human being. It's compact bone. Yeah? So I have to tell you, sorry, I don't know. How it's processed. Is our blood made in our bones? I find that fascinating. Yes, blood is also the white blood cells, for example, are made in the bones, in the hollow bone, so basically in the bone marrow. It's very important. It's very important for the immune system. Yeah. But again, I have to tell you, with the question, how was it stained? How was it processed? You're kind of speaking out of my heart, because I don't know, and I myself would like to know this. Here we see it much better. Do you see those little lines that radiate outwards? These seem to be the blood vessels. I did a little bit of research before today in the afternoon, and these seem to be the blood vessels that actually transport the blood also outwards. And I said compact bone because the second type of bone that there is called spongy bone, and so I don't have bone here with me, but the outside of the bone is compact bone. It's very hard, and if you look at the cross section into the bone, then you're going to see it looks more like a sponge, because bone also has to be light. And the large bones, like an upper arm and so on, they are even hollow inside, and where there is the bone marrow. So, but I think one of the important things here is that a bone, of course, is it's kind of obvious, but it kind of makes it clear here, is a living structure. Bone has to grow, obviously. And this means that there have to be cells inside the bone. And of course there has to be, blood supply as well. Okay, and this is basically what we see here. So this is already pretty high magnification. It's a 60 times, but let's go down a little bit so that you get a better overview. Okay, and there's 40 times magnification here. I think the arrow is pretty nice. Here as well. You see this at the bone. There's always this canal in the center, and then there are the bone cells arranged around it. So that's kind of here you see also a little bit better how the blood vessels are radiating outwards. From this central version canal, I think it's called. Osteocysts, that's how these cells are called. And of course there is the bone. There's of course a lot of minerals are also deposited, because the bone has to be hard. So let's go down with the magnification a lot, and then I'm going to move on, because I would have plenty of slides to show you as well. But I think when we get this overview here, then we can actually see this. Let me close this a little bit. This repeating structure, where you always have those circular arrangements, and always blood in the center. This seems to be some kind of a, I don't know, dust fiber here. So this is indeed a human tissue, okay, according to the label. So let's move on, okay. Occasionally I look over how similar to a tree it looks. Nature seems to have a pattern. Well, I would say that there are certain analogies and similarities, because if you want to transport, of course, let's say water or nutrients inside a plant, then you of course also need canals. And there are certain physical laws that kind of apply to all living things, and which kind of determine maybe also the shape of certain structures. So the arrow is nice. It's easy to follow. Then when you're explaining, yes, I'll tell you a little bit about this arrow. There's a little story behind this arrow. I've made the arrow myself in a PowerPoint first, and then with graphics. I did a lot of research online on how you can make a moving arrow in this program. And I was thinking about maybe you have to download some additional software and complicate it and everything. And I could not find any information, and then I realized why. Because there is no need for anything. You just put a piece of graphic over it, and then you can move it around. That's why it was never explained, because it was already included as a feature anyway. So it was, Sometimes it's so much easier. So this is basically the first thing I wanted to show you. So it's a little bit arbitrary. Yeah, this here is I can talk a little bit more about this here. Testes of a mouse, the testicles. Okay, it's quite interesting here. Yeah, I have to switch over. So these are the testicles of a mouse. Remember, the testicles are the places where sperm cells are made. Okay, and let's have a look here. What we have here are the cross sections of so-called the seminiferous tubules. So that's it looks like a circle here, yeah. But in reality, that's the cross section of a tubule. So you have got in the testis many tiny tubules running, and I can even talk a little bit about this here because this is actually one of the topics that I have to teach in school to my high school students. Okay, when you start to zoom in, okay, then you see that this is a seminiferous tubule over here. Yeah? And this is a fairly thick wall of the seminiferous tubule. And the inside here is the lumen. A lumen is like what we call an empty space. Yes? Yeah, and when we zoom in yet further, then we're able to see the individual cells much better. Okay, so this is again a seminiferous tubule. And what you see inside here, maybe you are able to see those fine little lines in the center, in the lumen over here. These are the tails of the spermatozoa, of the sperm cells. So remember the sperm cells, yeah, they look a little bit like a tadpole. They have a very long tail that moves. And what you see in here is there are the tails of the finished sperm cells. So this is the wall of the seminiferous tubule. And what you have on the outside here, this is referred to as the basement membrane. Maybe I find a better one over here. And I'm going to tell you now how sperm cells are produced. And by the way, yeah, these are the testes of a mouse, but it looks the same in all mammals. So human testes in the cross section look the same. Here you see those. Do you see the lines here? Okay, these are the tails of the sperm cells. On the outside here we have essentially it's called the basement membrane, and then there is a layer of cells right beneath it called the germinal epithelium. I think you don't have to know the terms here, I'm just saying it. Yeah? And these cells over here, they start to divide. And what they do is they undergo cell division. And as they divide, the cells move inwards until you have the finished sperm cells in here. And then the sperm cells, they will detach and then they're carried away. Yeah? So essentially, the sperm cells originate, or they start off being made here in the wall of the seminiferous tubule. Yeah? And the cells divide, they undergo a process that we call meiosis. And then from one cell that you have here, from the germinal epithelium, at the end you make 4 sperm cells, and those sperm cells, or spermatozoa, are then found in here, and then they're transported away. Yeah? So I think I personally like the cross section of the testes as a microscopic specimen quite a lot because it looks kind of good. Here, much nicer. Look, do you see all of those lines in here? It's much more organized here. Yeah, here, that's a nice one. Okay, that's actually really nice. Let's go up yet further. Yes, here, that's a good one. Up, I changed the light a little bit too much. So let's focus. Yes, this is a good one. Okay, look, I'm going to explain this to you. Okay, so you have the germinal epithelium layer over here. The cells divide, they undergo meiosis until you have 4 sperm cells per starting cell. And what you have over here, these are the heads of the sperm cells before they detach. So that's basically the head of the sperm cell, and then there's a long tail. Yeah. You see hundreds of them in one seminiferous tubule. And as I read somewhere, a human man makes approximately, I don't know, 70 million sperm cells every day. So I think that's quite fascinating to see here. Yeah? That's again, this is I know a little bit more about this one here because this is something that I have to teach in biology. Another thing that you see over here, notice over here, is these are the so-called the interstitial cells. Interstitial, no, the Leydig cells. Okay, Leydig cells they're called, and those Leydig cells, what they do is they produce those Leydig cells. They produce testosterone, the hormone, and you might also be able to see some blood vessels over here. It's a little bit red over here. Okay, and those yeah, blood vessels that carry blood. Okay, yeah. So that's basically also yeah, I think quite a nice specimen to look at. During the cutting process, some of those, you can see that some of those seminiferous tubules actually started to break down a little bit and start to rip and tear. You might wonder a little bit is if it's possible to make those slides yourself, provided that you have the tissue. And I said, yeah, but that's pretty advanced stuff. So to prepare to make histological specimens like this, it does require quite a bit of patience, a little bit of laboratory work, because you have to dehydrate the specimen, you have to embed it in paraffin, and then you need a microtome to make thin cuts, and yeah, you need to work with xylene, which is a solvent which is not very healthy. So and you have to stain it as well, because I think pretty much all of the slides here are stained. It doesn't say which stain, but yeah, and it's a pretty elaborate process. Here, very nice. That's a blood vessel over here, obviously. Yeah. So, yeah, let's move on. So these were basically the testicles of a mouse. Now let's see what else we have here. Let me remove the slide again. Up. Okay, and what else do I have? I just put the slides away here. This here is the lung of a cat, Felis domestica. Yeah, and you're able to see the so-called alveoli, the little air sacs. So I'm going to explain this as well. And by the way, in the upcoming weeks and days, I'm going to post a video in my other channel, in the main channel, where I am dissecting the lung of a pig, because a student of mine brought it along, and then I'll also be talking a little bit about this here. So when you look at the lung of a cat, again, it looks pretty much the same also for humans. You're going to see there's a lot of white stuff here. Let me go down with the magnification. And you see that there's a lot of, lumen, yeah, the white stuff. And then there are some fairly thin walls here of cells. And this kind of already illustrates that the lung is a very spongy in nature. However, it is not completely hollow. There is a lot of blood in the lung because the blood vessels over here. Yeah, but in here, these are the so-called the alveoli, or the so-called the air sacs. And this is when I inhale, breathe in, then of course those air sacs, they start to expand, and then there is going to be gas exchange happening. So basically carbon dioxide gas from our blood, which is here on the side, will go into the air sac. It will diffuse into the air sac, into the alveoli, and the oxygen in the air will go into the blood. Okay, and yeah, so let me go up a little bit and let's have a closer look at the walls of these alveoli. Yeah, so you see that these are the dark dots, those dark dots that you see, these are the nuclei of the cells. Okay, so yeah, the cell of course is much larger, the cells of course are much larger, but the nuclei, they stain darker because very often the company that made those slides used stains that stain DNA. And the DNA can be found inside the nucleus. So you see all of those dots here are the nuclei. And look at this. This red thing here looks a little bit like a blood vessel. Okay, yeah, so red blood cells are here. And you see that the walls of those alveoli are fairly thin. Okay, just a few cell layers thick. Yeah. So this actually means that the oxygen and the carbon dioxide, they have very short distances to move, the diffusion distances. Yeah. And you can also see that the overall surface area is quite large. So this also seems to be a blood vessel over here. Okay, possibly red blood cells. Yeah. So yeah, so very nice to see the thing is with those histological slides, what we have is we see only a two-dimensional cross section. In reality, the alveoli, those air sacs, look a little bit, are sometimes compared to grapes. Okay, they are, yeah, of course not two-dimensional, but they're three-dimensional, like little bubbles, like little grapes. Yeah, and basically because they're cut, therefore we only see a two-dimensional cross section here. Yeah? So you see the overall surface area is pretty large. And you also see how delicate actually everything is here, yeah, because it's only a few cell layer is thick. Again, seems to be a blood vessel over here. So, yeah, let me, this is pretty high magnification, but sometimes you can, okay. By the way, look over here. Can you look over here? Look at the light. What happens to the light when I switch magnification? It's a nice feature of a microscope. Look. Yeah, do you see that the light changes? I can pre-program different light settings for the different magnifications. Yeah. Here we see it as well. Yeah, that's seems to be a blood vessel as well here. Or could it be? Yeah, it could be. Yeah, because it's red inside, so these must be the red blood cells. But here we see the spongy. Yeah. Nature. The question is how large would they be, the alveoli in humans? They are microscopic, as you can see over here. But what I read is that there are several million alveoli in one lung. There is actually no reason for the alveoli to be significantly different in size. So I would say that, because of the physics of gas exchange and so on, I would probably assume them to be pretty much the same size. So basically, just because a cat is smaller does not necessarily mean that all of these microscopic structures are much different. Here again, a blood vessel. I'm going to look at a blood vessel later on again. Yeah, but here you see the red blood cells inside, and here the wall of the blood vessel. Okay, so let's move on again. So I've looked at 4 slides now. I know that this live stream is a little bit different. The other ones were a little bit more practical, but I said, why not do something like this as well? Artery and vein of a mammal. Okay, this one is also a very common slide that I show to my students. Let me switch over again over here. Always start with a low magnification first. And again, we have a cross section, okay? Let me go down yet further with the magnification. Okay, I need to flip over the, here we go. Okay, so this at first sight might seem to be a little bit confusing. Where are the arteries and where are the veins, and what are the differences? Okay, as a matter of fact, you see both of them here. Arteries are those blood vessels that carry blood away from the heart, and veins are those blood vessels that carry blood to the heart. Okay, so that's why we have a blood circulatory system. And those blood vessels that carry the blood away from the heart, the arteries, they have to withstand a very high blood pressure. So for example, you probably all know it, if you feel your pulse, okay, here, then that's an artery. And you are able to feel it pump here because of the changing blood pressure. I mean, some of my students always thought, and even I myself used to think that the pulse that you feel, that's because the artery kind of continues to pump the blood, but that's not correct. It's simply a result of the blood pressure changing. Okay, every time when my heart beats, the blood pressure rises, and I'm able to feel that. And because arteries have a high blood pressure, therefore they must have a thick wall in order to withstand the high blood pressure. And what we have over here is an artery, the cross section. And here in the center, in the lumen, in the white part, this is where you find the red blood cells. And you see that the wall is pretty thick. Okay, it's now collapsed. It's kind of pressed together. That's why it looks so narrow. Okay. But actually, when there's blood in there, it's of course round. So this is the 4 times. So let's go up a little bit with the magnification. Okay, so you again see that the wall of the artery is very thick to withstand the high blood pressure. And not only that, the arteries, they also contain so-called smooth muscles. Yes, there are muscles in the arteries as well, because these muscles are able to contract to regulate the blood pressure. Okay, yeah. So there's a question again: where do you buy all of these slides from? Well, the easiest way to buy slides is I bought this from Amazon. Okay, so this is one thing, and there is indeed, and I'm going to put a link to a catalog. There is a company in Germany a well-known one, who sells these slides here, and you can even buy them individually. Yeah, and they have hundreds of slides. The slide boxes, the slide boxes here are general purpose slides that contain an assortment of plant and animal tissues. But if you really want to have something specific, okay, for example, because you're for medical training, for example, then you can buy those slides as well, okay? So I would say if you don't have slides yet, and if you just want to have something to look at, get yourself a reasonably cheap slide. I don't know, the price also varies from Amazon, but I would say that I do have to say that some of the slides that I bought from Amazon cheaply are not quite as good in quality as the other ones I have here, because those slides here are also used by sometimes for medical training. Okay, so, and some of the slides can be quite expensive, and I checked the website of the company, and they said that for some slides you actually have a waiting time because they have to manufacture them and make them. Yeah? And I remember also that once I went to a local to a shop where it was actually a microscope shop, and they basically said is that they also have slide boxes just for about human anatomy of all different types of tumors. It even goes that specific. Yeah, so actually for med students, for medical training, so this is the thing. Sadly, the slides bought were dirty. Yes, this is I have, that's one of the quality aspects I'm talking about. I mean, if you want, I can have, we can look at some of those as well. And it's yes, they were dirty, and some of them depends on the company and on the quality of the slide, and not just dirty on the outside, but actually dirty on the inside. So where there is the permanent mount. So there is a price difference and there is a quality difference. However, I would say, it doesn't matter. If you don't have a slide box yet, then just get one, simply to have something to look at, if you're interested in it. So that is an artery, okay? So let's go back here. In here, you should be able to see the red blood cells. And you're gonna ask me, okay, where are the veins? The veins carry the blood away from the heart. And the veins carry a blood of a low blood pressure. So yeah, I can also show that to you. I mean, it's difficult to see here. I mean, it's difficult. Sometimes you see blood vessels on the surface here, okay? And these are difficult now. I don't have so many right now, but blood vessels that you see on the surface here, these are veins, and they carry blood of a low blood pressure back to the heart, and therefore their wall does not have to be so thick. And look, it's almost difficult to find, but this here, let me center it. Do you see this here? This is the cross section of a vein. It looks like a number 2 in this case because it's collapsed. And look how thin the wall is. Okay, so artery over here, vein over here, and it's very, the wall is very thin, and here it's collapsed. Yeah, and it's so thin because it does not have to withstand a very high blood pressure. We can zoom in again. See, even though this is actually one of the better slides, you see that there is also a little bit of dirt here on the slide. But actually, I've seen worse ones. And in here, these might be some red blood cells. Is blood taken from veins darker in color due to the lack of oxygen? I heard that as well, that there seems to be a slight color difference, but I think apparently, but if the blood comes out, I gotta do a little bit more research on this. I did hear that there might be a slight color difference in the blood, but if you take it out, then because as soon as it gets in contact with the air, it starts to become enriched again. But I would say, I'm not so sure about that. Okay. This would be an interesting question. Normally, when you have diagrams, you always paint blood that is lacking oxygen. You paint it in blue to indicate that it's rich in carbon dioxide. But in reality, of course, the hemoglobin is red. The question is if oxygenated hemoglobin is a little bit brighter than deoxygenated hemoglobin. Good question. Got to do a little bit of research on that. Okay, so So let's have a look again. So this was basically the artery and vein. Okay. There's, I just want to say one brief comment a little bit. I made a video recently, also in this very channel, if you go back a few days. I showed those slides to my nephew, 6-year-old nephew, and my sister-in-law. And yeah, I simply wanted to show them a little bit stuff under the microscope, and I showed them some of those slides. And actually for her, she said something quite interesting. She said, but those slides, they all look the same. They all look so two-dimensional, so abstract. And indeed, that's the case. And I think that would be that's a little bit of a disadvantage of those histological slides, is that in many cases there is no description with them. So basically, you're looking at things and sometimes you don't even know what you're looking at. So, and that's a little bit of a, I would say something, a little bit of a pity. So this is the scalp of a human. So that's a human, scalp is the skin of the head, cross section, and with the hair roots. Okay, so let's have a look at this because it's also quite nice to look at. All of these slides are stained. Yeah, here we see it quite well. Okay, but let's try to make sense of what we see over here, because some of the parts we already know or we can imagine. So I can imagine that, let's have a look. Some of the structures, like these could be maybe some cross sections of blood vessels. But look at this. What are those oval structures here? Those round structures. I want to have a closer look here. Okay, let's zoom in a bit. Okay, and I need to refocus because I think I'm focused now a little bit on the dirt. Okay. And if you look carefully, and if we go in yet further up with the magnification, we're going to see that, let me focus, where's the focus here? Not so good. Let me open the condenser a bit, go down with the, and refocus. I should have cleaned the slide a little bit. But what you see is the following. You see over here many cells, okay? And this is a so-called the hair follicle. Okay, you see a lot of cells in here. Some are a little bit darker, not those big dark dots. These that seems to be indeed a little bit of dirt and dust. Just a second. Oh gosh, forgot the condenser. Okay, here we go. I forgot. I removed the condenser, and so that's much better now. Okay, so look at all of those cells here. Okay, so those dark dots again seem to be the nuclei. Okay, it's so-called the hair follicle is the place where hair is made. And then over here, if you look at this red structure over here, I don't seem to see any cells, but rather it looks kind of fibrous. Let's go up a little bit with the magnification here. Okay, yeah, here now we see it quite nicely, the individual cells over here. And here I don't see those cells, but rather it looks kind of fibrous, and I think that this is actually the hair. Actually, I'm quite sure, certain about this. Yeah. So those in the hair follicle, those cells, they are producing the hair protein, and it looks like this because it was cut very thinly. And you see also a little bit that it seems to be broken a little bit here, because of the protein which was so hard. Yeah. It seems that a couple of different stains are used. Yes, most certainly. I don't know, again, I don't know which ones. It's a pity, yeah. But I'm quite sure that this is stained, I don't know, maybe even bleached, so that the hair color, the original hair color is gone. Yeah, could be, and then it's stained. And different types of stains, they react with different substances. Yeah, maybe parts that are more fatty or parts that contain more proteins. Yeah. But this again seems to be the hair follicle, and I don't know, but this a little bit looks like the cross section of muscles. We're going to be looking at muscles as well. Yeah, but those bundles that you see over here are like cross sections of muscles. Yeah. Yeah. So why muscles? Because there are also muscles attaching to the hair that are able to raise and lower the hair. I mean, for example, when you're feeling cold and the hair, they start to stand up. Here again, you can see the individual cells, but over here I do not see maybe this is a little bit of a part of the hair being formed. Yeah. It seems to be cut diagonally. Yeah. So if you go down with the magnification again, okay, and then we get also a better overview. So this does look a little bit like muscle. Okay, over here, muscles. As well, but that's kind of interesting because if they were both muscles, this looks also like muscles, but if they were both muscles, they actually should stay in the same way. Yeah, so, not sure about that. But later I'm going to show you how it actually looks like. Yeah. Could be a gland, maybe, yeah, with a canal going into the hair follicle. Because oil gland maybe. Yeah, and here are the different layers of the skin. Yeah. Yep, so most certainly different stains were used here. Okay. So let's have, where's the arrow? Let's have another look at something. What do we have next? The ovary of a cat. Yeah, I'm going to show this to you. I'm not quite happy with this specimen here because the follicle, is the part in the ovary that makes the egg cell. I've seen better specimens. I'm just going to show you anyway. Yeah, ovaries in a female cat. The ovaries are those parts that produce egg cells. Okay. But again, I'm not so super happy with this one over here. Okay. What we're able to see again in the ovary. Let me put this. Okay, so this is basically the complete structure. In a human, of course, the ovary is significantly larger. A few things that are already quite familiar. You see those round structures here, cross sections of blood vessels, which we already know. Okay. And then what you normally see is you see bubbles, and it could be that this is one of them, and those bubbles are the follicles. And let's just assume that this is one of them here. And inside the follicle, what you have is you have the egg cell ripening. Okay, it's a little bit too bright now. Okay, here let's go up with yep. It could be that this is a follicle. I've had another slide. I don't know where I've seen this, where the follicle of a rabbit, or the ovary of a rabbit, which is again a little bit bigger, but I've seen that there were several of these follicles because several egg cells were being produced at the same time. Yeah. Yeah. So this is an ovary, again blood vessels here. Okay, and if you zoom in, what do we see again? All of those dots, cells. I mean, if there's any take-home message, anything that we learned from this here is that, of course, living things, humans, mammals, animals are made of cells. Yeah, it's kind of really interesting to realize that it took quite long for people to actually come up with a cell theory, which states that all living things are made of cells. Yeah? It were two scientists, Matthias Schleiden and Theodor Schwann, two German scientists back in the day, in 19th century. They basically independently came up with a cell theory, or collectively rather. Schleiden said all animals are made of cells. Well, here we've got the cells. Schwann said all animals are made of cells, and Schleiden said that all plants are made of cells. Yeah, and I think it's already quite understandable if you look at these things, simply to realize that these little dots here, all of these little circles, are cells. Maybe it also took a little bit of time to come up with appropriate staining techniques. Okay, might also not be easy. Yeah, so there are, yeah, and again, lots of cross sections of blood vessels here. Yeah, yeah. Again, I've seen probably better yeah, examples of an ovary. And here on the outside, always lining the surface of an organ, you always have the so-called the epithelium. The epithelium are always those cells that are lining the surface of an organ. Okay, so let's move on a little bit. Okay, and what is this here? This one is, Skeletal muscles. That's an interest. That's in other words, skeletal muscle is meat. I mean, the meat that we eat is muscle. And let's have a see if we're able to see the striations, because this is a little check on the quality of your microscope. Okay. Skeletal muscle is also known as striated muscle. Here it's a little bit out of focus again. And we really have to go up with the magnification here. Okay, you see all of the individual muscle fibers here. Yeah, running across. And let's go up and let's see if we're able to see the so-called striations. Yeah. Do you see those? Here it is. Do you see those little lines running in parallel? That's why skeletal muscle is also known as striated muscle. Let's go up to 60 times. Nice, huh? Very characteristic. The second type of muscle that is the so-called the smooth muscle, which does not have these striations. And then you have the cardiac muscle, the heart muscle, which is the third type. And those striations are called the light and the dark bands. They're actually called like that. And they're because of protein fibers. And the protein fibers, what they do is a muscle contracts because those protein fibers, here we go, they kind of slide past each other. Okay? And depending on whether the protein fibers overlap or not, this gives, and the type of protein fiber rather, not whether they overlap, but the type of protein fiber, there are two different types where they are, gives you the light and the dark bands. And these are basically the striations. So the protein fibers, they run lengthwise like this. Oh, they run, here's the arrow. The protein fibers, they run lengthwise, and then the muscle is able to contract this way. So this is a little bit of a, if you have a slide like this, is a little bit of a microscope check if your microscope is able to resolve those tiny striations here. And here you see it again. So, yep, so that is basically skeletal muscle. In this case, actually from a human being, at least if you want to believe the slide label. But it doesn't really matter, because I have also a slide of a flea, of a dog flea, and there you can also see the striations in the muscles. Yeah? This one I think must also have been stained by some stain here, because we see the different colors here as well. Okay. So what time is it? Okay, 3/4 of an hour already. A few more. Yeah, I've got here another one. This one here is again a cat, the eye of a cat, the retina. Yeah, I'm also semi-happy with this sample here, because yeah, I'm just going to show it to you. So this is now the eye of a cat. It might not really look like an eye to you at the first sight. So but let's try to make sense of the things that we see here. Let's start again with a low magnification. Okay. Let's have a look here. Yeah, I need to flip out the, here we go. Yes, look at this. Look, this might be the thing here. So what we have is this is most likely it is the retina. This is the layer, the inside layer of the eye, which is light sensitive. And I think, look, it's ripped here because this happened all during the preparation. Okay, the retina also seemed to have separated here, yeah, from the rest. Yeah, the retina separation is also, as a matter of fact, a disease, a problem. Yeah, and but I think here it might have separated because of the preparation. Okay, and you see also over here that there is a dark layer, and I think, again, correct me if I'm wrong. Maybe some of you know. This seems to be the layer called the choroid, which is a darkly pigmented layer which reduces internal light reflection. So what you have is in here, this inside part here, that is the inside of the eye of the cat. Okay, and the light is projected onto the light sensitive layer here, and over here on the outside we have muscles again, and that's the outside of the eye. Okay, but let's zoom in a little bit. Let's go up with the magnification. Yeah. Okay. You see that, yeah, it must be either this layer over here or this layer over here, or maybe it's this one. It's a darkly pigmented layer called the choroid, which reduces internal light reflections. And one of those layers here must be the light sensitive photoreceptors, the nerve cells. Yeah? But essentially, the inside, with my students, I always dissect eyes, pig's eyes usually, because we can get them quite cheaply. And when you open them up and when you cut them open, you're going to see that the inside is completely black. The reason is that you do not want to have any light bouncing around. So for example, also the inside of a cinema, if you go to a cinema, for example, then you are also going to see that the walls generally are quite dark because this increases the contrast. Yeah, so again, here the retina, very nicely visible that there are different layers with different responsibilities. And a little bit unhappy I was because I was not able to see quite well. I think these lines that you see over here might be the axons of nerve cells. Okay, let's go up a little bit. The axons are those part of a nerve cell that carry the signal away. Yeah? Again here. Again, that's one of these things where we, it could be that one of these here might be the photoreceptor layer. Then there's a ganglion layer, maybe that's the one over here. Yeah, and the inside somewhere, maybe either this one here or this one over here might be the layer of darkly pigmented cells, the choroid, yeah, which reduces the internal reflections. Yeah. So actually the photoreceptor layer could also be this one here because it has, the light has to first pass through the so-called the bipolar layer and the ganglion cells before it's able to reach the photoreceptors. Yeah. Yeah. There's another question here. Are you moving out your condenser to get rid of the vignetting with a low power objective? The answer is yes. Okay, I'm going to show you. That was actually one of the questions that when I got the microscope, I immediately called the company. Olympus, why don't I get the full field of view? Okay, look at this. Okay, so this is the low power 4x objective, and this is the Köhler diaphragm. But you see that on the side here, yeah, it's vignetted. It's dark. Yeah. So what I have to do is I have to swing out the condenser. Look what's going on here. Okay, look at this. Yeah. Up. I can swing it out. Yeah. And then I'm able to get the full view. Yeah. 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 So that is, yeah. But if I leave it out and go up with the magnification, then it's not quite as bright. So I have to swing it back in again, and then it bundles light better. Okay. Yeah. So well observed. Yeah. I was a little bit when I first got the microscope, I was a little bit confused. And then I realized, I was told that actually you can swing out the condenser and then you get the full view with the low power. Yeah. I think this has some kind of mechanical or technical reasons. Yeah. So what is next? This here is now the small intestine also of a cat. Okay. Yeah, so let's have a look here. We are actually able to see the infoldings of the small intestine. So let's see. Here it is. Again, fairly thin. So let's have a look here. The inside of the small intestine. Okay, and notice this here that it is not smooth. Yeah, so there are infoldings, they're also called the villi, and this increases the surface area. So the food being digested is in here. Okay. This is the outside of the intestine. There must again be an epidermis or epithelium, on the surface here. And the inside here is, actually has many finger-like extensions and is not smooth. And this is to increase the surface area for absorbing the digested food. So let's move again here. I need to refocus. Okay, very nicely visible. Okay, that the inside is, has those extensions to increase the surface area. I don't know what these things here are, if there's some kind of preparing artifacts. Yeah. Poor kitty. Yeah. Yeah, there are companies that make those slides. Yeah, so I know, poor kitty, but at least you can make a lot of slides. You can make a lot of slides from one cat or from one organism. Again, over here, those infoldings. Yeah. And then there are many blood vessels in there as well, which then transport the absorbed food away, yeah, to the rest of the body. Here, see, this also seems to be a blood vessel over there. You must have used again a stain that stains the DNA so that the nucleus, the nuclei are more visible. Yeah. I hope they use cats that died naturally. Yeah, or maybe, yeah, maybe they put some. I think especially if you're working, yeah, as a veterinary doctor, I think they have a lot of specimens available. Yeah. Yeah. So if you have to put a cat to sleep, yeah, basically if you have to mercy kill it because maybe of old age, yeah, then I can imagine that, yeah, they might use some of the organs. Yeah. Yeah. It is fairly easy, under quotation marks, to use a microtome to create thin sections from a plant. How would you go about creating a slide from, say, meat from a butcher? Is that even possible from an amateur? Yeah, I can tell you a little bit about this. So basically, how do you make slides, permanent slides, and cross sections from tissue, from animal tissue? Okay, so that is that's the question. Very briefly, there has been a question: where's a blood vessel? The blood capillaries here are too small to see, but over here somewhere I've seen a blood vessel. Yeah, somewhere. Where is it? I'll find one. Yeah. So basically, yeah, here, that's a blood vessel, for example. Yeah. So the question is, how do you make those slides? I can briefly tell you, at least theoretically, because when I studied and so on, we had to read up on this and know this a little bit. Is you have to take a piece of meat, let's say muscle tissue, whatever tissue, and you have to first of all fix it. This means that you have to put it into a solution which denatures the proteins. You have to keep it in there. Yeah, I think they call it a formalin or solution, and this kind of infiltrates the tissue and conserves it. And after the tissue has been conserved, you have to take the water away without causing the tissue to shrink. And you do that by placing the tissue into an alcohol series. So that means you slowly go up with the alcohol concentration, and you leave the tissue in this for several days. Okay. It is formaldehyde or formalin. One of those two is dangerous. The other one is used commonly in preserving. I don't know which one. One is highly cancer causing and is not allowed. And either it's formalin or formaldehyde. Yeah, you use the not so dangerous one, obviously. Yeah? It smells pretty badly anyway. Yeah? So it's formalin or formaldehyde. I forgot which one is used. But one of them is pretty dangerous. And then basically you have to dehydrate it. And this basically means that you increase the alcohol concentration until all of the water has been removed. And you have to increase it stepwise so that it doesn't shrink. So you have to give the alcohol some time to replace the water. And then after you've used ethyl alcohol, then you have to transfer to isopropyl alcohol because this contains even less water. And then you have to transfer it into a solvent called xylene. And xylene is again a solvent which is not so healthy. That's why it's not recommended for home use. And after you've put it into xylene, you have to then put it into paraffin. Paraffin, which is a form of wax. And then you put it into wax blocks, and then you can cut it with a microtome. And then you have to again treat it with xylene, in order to remove the wax, the paraffin. So you see, it is a pretty elaborate process, and for this reason, it's I would say not so many amateurs do that, but I've heard of a few people who actually do. Yeah. Formalin is a diluted formaldehyde in a buffer solution. Interesting. Yeah. Yeah, I'll leave it to professionals. Yeah, I think botanical sections are easier. Yeah, I have to. Yes, I agree. Yeah, botanical section, you take a stem and you take a microtome, and you make thin cuts, and you'll be fine. And animal tissue is indeed more complicated. Yeah? So let's move on a little bit. I always have to go down a little bit with the magnification. What else do I have here? I'm already finished. Well, I do have a few others here as well. I mean, there are a few things. Pig, motor nerve. Yeah, I can show you some nerve cells over here. I'm not so happy with this slide here. That's why I put it away. Because actually I was kind of hoping to be able to see the individual nerve cells, the individual neurons. But this is difficult here because they're all so together. But there is one thing that you are able to see a little bit occasionally. Okay, so that is supposed to be nerve. Okay. But where I want you to direct the attention to is the following. You're going to see that sometimes there are those horizontal fibers running. Okay, you see it's not, there are some lengthy fibrous structures. Yeah? And these seem to be the so-called the axons of the nerve cell. Axons are basically a nerve cell, looks like this, that there is a nerve cell body and then there's a long extension called the axon. And this is where the nerve impulse is carried. And I was actually hoping to see them a little bit better here, but there seem to be so many of them bundled together and they're so thin that it might be difficult to see. But you do see that there are these lines kind of running, and this seems to be where the axons are. So there's another comment here. The International Agency for Research on Cancer classifies formaldehyde as a human carcinogen. Okay, yep. So that's, yeah, here as well. You see those long things here? Yeah. So there is another question. Would you buy antique prepared slides from eBay? What I heard is that at least if the slides are collector's items, especially from the 19th century and so on, they can be quite expensive. Okay, the thing is I know that those real antique slides, which are also sometimes have those paper, they're wrapped in paper and they look kind of artistic, seem to be collector's items. So would I buy them? Why not? If you want to have them. I don't know if you were to buy them primarily for the specimen, or do you want to buy them primarily because you want to collect antique slides? So I think that is the question. If I basically, you see, if I'm in education as a teacher and if I want to do microscopy with a whole classroom where you look at different specimens, and I have to make, I need a large number of slides, which basically are very similar. So for example, when I look at the blood vessels, the artery and the vein, pretty much all of the slides in the slide boxes, they look the same because it's from the same series and they're just cutting with a microtome, and so basically the slides are all very similar. Yeah, and this makes it kind of easier. So if you need, for education, I would not buy antique slides because it would be kind of a pity to use them with students, and I need more of them, And so I would say it depends a little bit what the objective and the purpose is. Yeah. So wow, it's already an hour, maybe. Yeah, and I just want to show you over here. Okay, I need to change again. Maybe I can. Yeah. Show you over here what it has. I mean, here, this one at least, it has first all of the plants, okay? And then it goes over, and here are all the animal tissues. Yeah? Dog esophagus, human blood smear, dog skeletal muscle, pig motor nerve, that's the thing that we just looked at, the spinal cord of a rabbit, the testes of a rabbit, okay? The heart muscle of a dog, which wasn't so good, yeah? Dense connective tissue, honeybee mouth parts, honeybee worker, leg, hydra, and dog stomach. Okay, here's a dog stomach. I don't know, we can have a look at this here as well. Okay. You see here, I have to find it here again. Layers. Could be that, again, this here is the inside where you see that the surface area is very large. Yeah? Can you demonstrate an ocular micrometer someday? Yeah, an ocular micrometer is a little scale that you put into the eyepiece. And this scale that you put into the eyepiece is actually, yeah, but my camera is not connected to the eyepiece, so I have to somehow think of a system where I can actually look through the through the eyepiece. But just a second, let me quickly check something in the drawer below. Just give me a second. Up. Let me, just a second. I just want to show you something else, because this is with the micrometers is a question that comes repeatedly. Look what I got here. This is not an ocular micrometer slide. It's not an ocular micrometer, but it's a micrometer slide that I bought. Okay, and just because there was yeah, let's look at the spinal cord. Okay, we'll look at the spinal cord. I hope it don't remind me, but I just, because there was this question, I just want to show this to you here. Okay, very briefly, because this might actually be also useful. Okay, so just going to put this in here. And I've actually used this also to make scale bars for references, but I need to see, I have to find it. See, that's a little bit the problem. Just a second, let me here we go. Look, here, that's one of them. Okay, that's one of them. And there is a scale in here. Okay, and then when you take a picture of this, you can actually use this as a reference. Where's the next one? I don't even know where the next one is. You know what, I'm going down with the magnification here. Okay, I have problems finding these things. Just a second. I need to, yeah, here there is a dot of a defined size. Here is, yeah, this here. Yeah, and here's, that's also a scale. Okay. So yeah, so basically that's also something that that might prove to be useful. So you basically take a picture of this, and then you can measure out yeah, the sizes. Of course, you need to for each magnification you need a separate one, obviously. Yeah. Yeah. So yeah, just as a little comment for some of you who might consider this useful. So there was the question about the spinal cord, number 18. Let's have a look at this here, and I gotta find it. 18, where's the spinal cord? Okay, rabbit spinal cord. CS means cross section. Okay, so let's have a look here. Yeah, okay, lots of cells running vertically, I suppose, and so let's try to make sense of the thing that we see over here. It's too large, maybe. Let's swing it out. Yep, here we go. Okay, and very nicely visible. Okay, this shape over here. Okay. This is also yeah, typical for in human spinal cords. Okay. So what you have is you have yeah, nerve cells basically in this case now running out of the yeah, computer screen. Huh? So that's why we do not see the axons so much because they run out of the screen. Again, what appears to be blood vessels, I would say. Huh? Okay. Yeah, and I think just by looking at a structure like this over here, yeah, where so many cells are packed so closely together, where it's so difficult to see the individual cells, then of course it becomes a little bit more understandable why people who basically were not sure. I mean, is this even made of cells? Okay, where does one cell start when the next cell stop? There's another thing that I just wanted to mention here is the following. It took people quite a long time to actually figure out that bacteria for example, are able to cause diseases. And now imagine the following, that you've got infected tissue with some bacteria on it. Compared to the number of cells that are in the tissue, the number of bacteria are relatively low. And so it is actually quite difficult to sometimes distinguish, to see the bacteria, if there is so much other tissue present which kind of covers the bacteria up. And for this reason, certain staining techniques like gram stains and so on were invented, so that you're able to stain the bacteria so that you're actually able to see them better in comparison to the human tissue. Yeah? Can we see some neurons with a nucleus? I'll be honest with you, all of these things are neurons here. Okay, but the axons, they run kind of into the screen. Yeah. And therefore, and normally when you draw a neuron, you always have a side view. So and for this reason, it's difficult to actually see them, because I can imagine many of these things in here might be nuclei. Okay. But simply by the way that because it's a cross section, could these be also maybe some nerve fibers running outwards? Okay. But these are all nerve cells here. Yeah. Possibly that some of these are nuclei, but again, it's I would say difficult to see. Yeah. So, do I have something else here? Dog cardiac muscle was not so good. The motor nerve, skeletal muscle, human blood smear. There's maybe, let me see if I have something here in the other box. This is a slightly older box here. Yeah, these were skeletal muscle. Yeah, and then some insects over here. Yeah, so it's from the time everything was written with a manual typewriter. So this box must be already from the, I don't know, 70s or 80s. Yeah. So Blutausstrich number 12 is, that's human blood. Okay, maybe that's also something. But I would say, if you want to look at human blood, I made some videos, which actually where it looks much nicer. But this is basically a human blood, and you go in with the magnification, you're able to see the red blood cells. Yeah. But then I have to honestly admit that looking at fresh blood is so much more interesting, because you can actually see the flexibility of the red blood cells. I did some staining with methylene blue. I was able to see white blood cells. Here's a white blood cell. That's this one here. You see, this is very typical. The dark blue stain is the nucleus, and white blood cells have a lobed nucleus. And by the shape of the nucleus, you're even able to categorize it. Now, I don't know which one it is, but this is a very typical, so this must be a stain sample. Yeah, so this is a white blood cell. The universe, just a second. What's the blood vessel density? Is this an easy way to calculate it? Blood vessel density. Do you mean how many blood vessels there are? And this depends entirely where you're looking at. Seriously, because certain parts of the body simply have a higher density. And also where you have blood capillaries, for example, in the lungs, in the intestine, fingertips, you've got a lot of blood capillaries. I mean, you have a lot of blood vessels. Okay, so the density would be higher. Let's put it this way: the density where gas exchange has to happen must be higher. This is especially the lungs, where capillaries can also be found in the kidney, in the liver, and so this is the place where the density must be high. In brain, of course. Yeah. So, but how to calculate it? I don't know about that. Did I use an electron? Did you use an electron microscope? No, almost. This is the answer. I wanted to use an electron microscope because at the Botanical Institute of our university, they had a nice electron microscope, and I actually participated in a tour where they showed it to us. but they only allowed students who have passed certain courses to work with the electron microscope, because it is expensive, obviously. And it was also like this, that I was told that essentially if you do something wrong, then you can actually damage it. At least at that time, I mean, we were talking about the 1990s, when I was still studying, it was pretty delicate. And so for this reason, unfortunately not. I just remember that I talked with a researcher who was working with the electron microscope, because sometimes it was like this, that actually the researchers, you had to give them the sample to actually take a picture of whatever you want to look at, because sometimes they wouldn't even let the people to the microscope. You had to be a trained specialist technician to be able to use it. And he was kind of demotivated. He said he actually said, well, working with an electron microscope is like an air traffic controller in an airport, he said, because you're always sitting the whole day behind the computer screen, and no natural light, and just looking at the screen the whole time. And he said it's very exhausting. So he said he did not have a very positive remarks about his job. Still, still interested me though. You might try to build your own electron microscope. Seriously, I did indeed hear that some people have attempted to do this. So this is not such a topic which is totally out of the ordinary. Can have private individuals have electron microscopes if they can afford them? Well, let's put it this way, a lot has happened since when I was still at university. Okay, so I know that the electron microscopes have become significantly smaller now, but when I was at university, the electron microscope, there was actually one room completely reserved just for the microscope, and there was a separate room containing all of the vacuum pumps to make sure that the tube of the microscope has a vacuum. So it was a separate room for that. And somehow the whole microscope had to be shielded, because there was somehow a tram on the outside with electricity, and every time when the tram passed by, this kind of interfered with the electron microscope. It was that sensitive, because of the electron beams somehow, and so on. So I just want to say that can private individuals, I've heard of people who collected electron microscopes. Seriously, many years ago I got an email from one person who was actually collecting them because he was working with them professionally, and then after he retired he kind of collected them and kind of fixed them and so on. Why not? I mean, museums have them, certain technical museums, so it's not totally out of the ordinary. But I would say that it's probably not it's probably not something that is very common, I would say. So it's basically blood. Here, this is the white blood cell, so maybe we're able to find more. You was actually quite lucky. So the question, it was this blood vessel density. Is there a way to calculate it? So basically the question is if you look at some tissue, of course you can calculate it. I'm just going to invent a thing. Let's say that you look through let's say this rabbit spinal cord. Let's put this one in again. How many blood vessels per square millimeters? Why not? I see no reason why I shouldn't be able to do that. You just have to count them. Okay. First of all, the problem that I have is identifying them. What's a blood vessel here? Okay. So or cell density. It's also something that you might. I don't know. It's a little difficult to see here, but I mean, if you were to see the blood vessels, and then you can count them, and then you measure out from where to where is 1 millimeter, and then you have 1 square millimeter, and then you count how many blood vessels you have in there. Okay, that's the way that I would do it. And then of course I would not only rely on one slide, but on several, and then you have to calculate an average. And if the blood vessels run vertically, then I would say that probably the result might even be quite reliable. So that is, in this case you do need the micrometer slide that I showed you before. Then you somehow kind of have to, what I would do in this case is I probably would not do this on screen like this, but I would actually take photographs. and then print them out and then stick them together maybe, and then do this on paper, because then you can actually check it off and mark it off and write on the paper for all of the blood vessels or all of the cells or whatever you want to count. You can kind of on paper check it off. Yeah? That would be kind of the way that I would do it for practical purposes. Yeah? Because otherwise if you miscount and then you kind of bump into it and then you don't know where you left off. Yeah? I would say, yeah. How to identify blood vessels? Well, arteries and veins. I don't know if you joined later, but I actually did show you a slide, if I can find it over here. But it's actually one of the artery and veins of a mammal. Okay, here it is. It doesn't say which mammal, but that's the one that I just showed you before. And if you joined in late, then you can, of course, the video is available afterwards for viewing. So this here, for example, is an artery. This here, cross section again. Arteries have a very thick wall because they have to withstand the high blood pressure. Okay, and this over here is the inside of a vein. It's pretty collapsed. Okay, and you see that the vein over here has a much thinner wall because the blood pressure is much lower. So a few more questions and then I'm already talking 1 hour 15 minutes. I'm already feeling a little tired. How would you compare the optical quality of this new scope compared to your previous Olympus? Oh, it's great. The previous Olympus was already good. I would say the following. It's not so much the optical quality alone, because if there's a bit of chromatic aberration, okay, who cares? But what is really nice when you look through it is that you have really a large field of view. And that's the nice thing, okay? The large field of view, so it's more immersive in that sense. So the subjective impression that you get is better. Yeah, but that's a question of the eyepieces. And so in that sense, I like it a lot, yeah? Oliver, we don't want you to leave. Well, I'm not, let's put it this way, I'm uploading videos anyway, and this video can be watched anyway. But you know what, I'm just going to show you, because you were asking about the eyepieces. Look, I'm going to pull it out. I'm going to show you how the eyepiece looks like. I'm almost scared a little bit. This is how the eyepiece looks like, okay? That's pretty massive. And it's also, the diameter is much larger than the standard diameter. Yeah. So in that sense, it's when you look through it, it's very nice. Yeah. Yeah. And I would say that this is actually one of the reasons why it's one of the reasons why you get such a nice impression. Yeah. What stain do I use to see white blood cells? Yes, this is I've used methylene blue Löffler's solution, and I made videos of this. So it's actually quite easy. You take a drop of blood, you add a little bit of this methylene blue solution to it, and you mix it. That's important. You take a toothpick and mix it, and then you put a cover glass on top, and that's it. Yeah, it's pretty straightforward. For example, if you look in my other channel, if you watch the video where I put my contact lenses under the microscope, okay, it's just one of the recent videos in my other channel, in the main channel. I was actually able to see a few white blood cells on my contact lenses. I was kind of wondering, what in the world do they do there? But I think that they are kind of naturally present to kind of clean up possible bacteria that are on the eyes. That's my hypothesis. Yeah. And what I've done is, because I've got monthly contact lenses, I simply took them, disposable, right? So I simply cut them up and put them under the microscope and with some methylene blue. You watch the video, but the same principle applies to when you want to look at blood. You just take a small drop of blood, you add some methylene blue solution, Löffler's solution, which is a ready-made diluted solution, and you mix it and you put the cover glass on top, and you can actually see the white blood cells quite well. Yeah, it's pretty straightforward. So you know what, I'm gonna call it quits now. I hope that you liked the video. I know it's a little bit different than in the previous one. Sometimes I do some more practical stuff. Today I decided, okay, I'm just going to show you a couple of my slides here. to kind of cover all aspects of microscopy and biology a little bit. Yeah, I don't know yet what I'm going to do next week. Sometimes it takes me a few days to come up with some ideas. If you have any suggestions, please post them in the comments. And yes, by the way, another thing that I want to suggest, what I would simply, if you are interested, some time ago I made a Reddit channel, not a Reddit channel, a Reddit page. So reddit.com/r/Microbehunter, and I'm also posting my videos or linking my videos in the Reddit page. And if you want to post pictures, I don't know, maybe you were kind of repeating some of the things that I'm doing in this channel here, and you also want to share your pictures, you can of course post them also in the Reddit page. Okay, yeah. So this is also simply a suggestion that I have. Yeah, I'm gonna call it quits for today. Hope you liked it. I liked it in any case. See you again hopefully next week. Yeah, happy microbe hunting as always. Bye bye, all the best, and happy microscopy. Yeah, bye.